For those of you joining us now, my name is Luke Evnin, and I'm chairman of the Scleroderma Research Foundation. It's my pleasure to introduce Dr. Peter Merkel, Dr. Max Konig, Peter Maag of Kyverna, and Dr. David Shook of Nkarta, who will present today as part of our session on CAR T cell therapy and other advanced biologics in scleroderma. Dr. Peter Merkel is the chief of rheumatology and a professor of medicine and epidemiology at the University of Pennsylvania, where he directs the Penn Scleroderma Center. Dr. Merkel is an internationally recognized researcher and clinical expert in scleroderma, and he is the author of over 400 scientific publications. Dr. Merkel's research focuses on clinical trial design and conduct, outcome measure development, clinical epidemiology, genetic epidemiology, and biomarker discovery. Dr. Max Konig is an assistant professor of medicine in the Division of Rheumatology at the Johns Hopkins University School of Medicine. Dr. Konig's research interests include immunoengineering and elucidating mechanisms underlying the initiation and progression of autoimmune diseases. His lab works to develop novel precision cellular and protein-based immunotherapies for the treatment of autoimmune and rheumatic diseases, aiming to selectively eliminate autoreactive immune cells while preserving protective immune responses. Peter Maag is the Chief Executive Officer of Kyverna Therapeutics, with a track record of transforming organizations and more than 20 years of executive management experience in various healthcare and technology companies. He holds board and advisory positions at a number of different companies, including Phoenix Pharma SE, CareDx, Miromatrix, and the Personalized Medicine Coalition. Dr. David Shook of Nkarta was appointed their Chief Medical Officer in 2023 and leads Nkarta's global clinical development and regulatory operations. He has more than 10 years of clinical research and development experience and has authored dozens of scientific publications covering areas including the quantitation of minimal residual disease in acute myeloid leukemia, AML, stem cell transplantation, as well as NK cell and T cell therapies for hematologic malignancies, as well as solid tumors. Before we begin what will be a very interesting conversation, a quick reminder: please submit your questions at any time in the Q&A, and we'll address as many as we can at the end. And with that, Dr. Merkel, I'll hand the microphone to you.
Hi, I'm pleased to be part of this Scleroderma Research Foundation Patient Forum on CAR T-cell therapy and scleroderma. This is an important topic and an area of active investigation that I'm involved in, and I think this will be an excellent session. My task today is to talk about immunologic reset to treat scleroderma. Why do we think resetting the immune system may help treat scleroderma? Well, scleroderma is an autoimmune disease. Autoimmune disease means that the body is attacking itself. The immune system that usually helps us clear infections and may even treat other things is supposed to be protecting us, but sometimes it goes awry and we have the immune system attacking ourselves, and we call that autoimmune disease. Almost all patients with scleroderma have autoantibodies. Autoantibodies are antibodies, which are proteins usually we have to respond to a vaccine, for example, and then attack a virus, or other infections. Patients have antibodies against themselves. Most patients with scleroderma have an anti-nuclear antibody, or ANA, and many will have other antibodies, such as anti-centromere, or anti-SCL70, or anti-RNP3, etc. Inflammation is seen in many organs, and that is also a sign of autoimmunity. There are laboratory experiments, including with animal models of scleroderma, that indicate the immune system is significantly involved in the cause of scleroderma. There's evidence that drugs that suppress the immune system can help some patients with scleroderma. cyclophosphamide, also known as Itoxan, mycophenolate mofetil, known as Cellcept, tocilizumab, which is known as Actemra, and other drugs have been shown to successfully help some patients with scleroderma in some situations. And all of these drugs suppress the immune system. And all of these drugs are drugs we use for other autoimmune diseases, such as rheumatoid arthritis or lupus. So what if we hit the immune system really hard in patients with scleroderma? Not just a somewhat milder approach that we do with these drugs. Can we reset the immune system so that it no longer harms the patient? And would that work, and would it be safe? Because of course we need both efficacy, that it works, and safety. So what is myeloablation with autologous stem cell transplant? Myeloablation means destroying bone marrow cells. Most of the immune cells that we're talking about involved here in this disease originally come from the bone marrow. Not all, but most come from the bone marrow. Stem cells are early cells that can later differentiate or change into immune cells. So these stem cells, we all have them, and then they can become this type of immune cell or that type of immune cell. Therefore, the goal of this therapy is to wipe out the bone marrow of immune cells and then repopulate the marrow by giving a patient back their own stem cells. The idea of this is that we will get rid of the bad immune cells that are causing disease. We call those pathologic immune cells. And that we'll repopulate with new immune cells that will help us restore our immunity and help protect against infection, but will not be attacking ourselves and causing disease, in this case, scleroderma. This treatment of stem cell transplant after myeloablation or chemotherapy has been used for many years as part of treatment for many types of cancer, and it has been life-saving, and it has become much more routine and safer over the years. So let's talk about the journey a patient with scleroderma would go through if they were going to get myeloablative chemotherapy and autologous stem cell transplant for scleroderma. The patient comes to the hospital, and we mobilize their stem cells by giving them G-CSF. This is a drug that pushes out these stem cells from the bone marrow into the circulation, into the blood. Why do we want them in the blood? Because that's where we're going to collect them. So this helps boost that effort. Once we've done this and there are more stem cells mobilized, we collect them through a process called leukapheresis. Leuka is white for white blood cells, and these are all white blood cells. And what we do is we take the patient and they get hooked up to a machine through an IV. Blood comes out of the machine, collects the cells, and then puts the rest of the blood back in the patient. We then take those cells and they are processed in a laboratory very specially so that we can just get the stem cells and make them ready to go back into the patient later. Now the patient's ready to get their myeloablative chemotherapy, and this is in 2 steps. One, they get total body irradiation. This is actually radiation therapy to the body, except we know it is important to shield the lungs and the kidneys so they don't get damaged through this process. This is combined with IV chemotherapy with cyclophosphamide and something called ATG. Combined, this really wipes out the bone marrow. So now you've had radiation and chemotherapy. Then the patient is ready to get their stem cells back, and they just get an IV, and their stem cells that they were collected earlier, they get their own stem cells back. And the idea is that these stem cells will know where to go, and they do. They go back to the bone marrow, they start making different immune cells. They make B cells, they make neutrophils, they make all sorts of different cells that we need. But hopefully they are making them that are not the type of immune cells they had before that were attacking the patient. So we get rid of the bad immune cells and bring some new ones back in and make new ones and restore or reset the immune system in the patient. So there have been 2 major trials of myeloablation with autologous stem cell transplant to treat scleroderma, and there have been other studies as well, but I'm going to highlight these 2 important studies. First study was done in Europe. It's called the ASTIS study, and this took This involved 10 countries and 29 sites. These are very hard studies to do. And this included 159 patients with early diffuse cutaneous systemic sclerosis, meaning they had skin that was more involved than some other patients. And they had to have had pulmonary, heart, or kidney disease, and sometimes 2 or 3 of those. They were then randomized to get either myeloablation and stem cell transplant, as I just discussed, or 12 months of intravenous cyclophosphamide, or known as Cytoxan, they would get it once a month, which is also a method of suppressing the immune system. The United States trial was the Scott trial, and this was done in 26 sites in the United States, 85 patients, also with early diffuse scleroderma, also with pulmonary or kidney disease, were randomized to get the myeloablation and the stem cell transplant, or 12 months of cyclophosphamide. You can see these are very similar trials. The results were quite similar as well. This is from the European study, the ASTA study, and this is overall survival, because that was the main outcome. We want our patients to live longer. And you'll see here that the patients who received the myeloablation and stem cell transplant did live as a group, lived longer. You can see the graph on top is better. Going down is bad. That's not survival. However, you'll notice in the upper left, there was a little bit of a blip in the first year, which was a little bit worse for the patients who get stem cell transplant. And that is consistently known because the first year is the most dangerous part. This is something called event-free survival, where you include other things that could happen regarding scleroderma. And again, there was a benefit to the patients who got the stem cell transplant. You'll see that here in the American Scott trial, very similar findings. The numbers get quite small at the end, so things get a little wider. And you can see that similarly, the events-free survival was different. But again, bit of a, not as good in the first year, but after the first year, better. So, in summary then, myeloablation and autologous stem cell transplants for scleroderma. Is this a good idea? A bad idea? Well, the pros for doing this therapy in patients with scleroderma is that it's the only treatment for scleroderma clearly demonstrated to improve survival. We actually think that our treatments for pulmonary hypertension and interstitial lung disease also help improve survival in some patients. But this was a dramatic change that was shown in these trials. And for some patients, they get substantial benefit. Their disease gets better and they don't have a relapse. And they do so off of other drugs. There's an increasing number of centers capable of treating patients with scleroderma with this approach. This is what we do at the University of Pennsylvania for selected patients. However, there's some cons and warnings about this treatment as well. The first-year mortality is now better than it was, but it still exists, and that causes some pause for some patients, as it should. The treatment is quite toxic and has some lasting effects, potentially. And unfortunately, it doesn't always work. It doesn't always stop progression of disease, or it stops for a while and then patients get a relapse later and have to go back on drugs to suppress the immune system after having gone through all this. Additionally, these trials had highly selective patients, and we are highly selective about which patients that we will give this therapy to at our centers. So many patients don't qualify. They're too sick, or they have other problems, other medical issues that make it not a good idea to give this therapy. It's also expensive. This is an advance in the treatment of scleroderma, myeloablation and stem cell transplant, and it represents a lot of fine work by investigators and effort by patients around the world. However, we can and must do better for patients with scleroderma, and I think we can. So why consider CAR-T19 treatment for scleroderma? CAR-T therapy has been used for a decade to treat and in some cases cure patients with lymphoma and leukemia. CAR-T cells eliminate B cells extremely efficiently. They really wipe out the B cells. B cells are immune cells thought to be an important part of the disease process of scleroderma. So there's growing evidence that rituximab, a drug that leads to B cell depletion, but not nearly as well as CAR-T cells do, has efficacy in treating scleroderma. So the hope is that CAR-T cell will, by just going after the B cells, will really reset that part of the immune system and be efficacious to treat scleroderma. So thank you for listening, and I look forward to answering questions later during our panel discussion.
Well, thank you very much for the introduction and for the invitation. I'm really excited to join today and talk about tomorrow's opportunities in scleroderma. There's palpably a lot of excitement about cellular therapies and autoimmune diseases, including in scleroderma. But when I think about the current generation of CAR T-cell therapies, even though the results have been transformative, we're facing four major barriers for these to get them into our patients. The first barrier is maybe a smaller problem for patients with scleroderma, but that is the use of conditioning therapy. These are typically used, as you're familiar now, to allow room for the cell therapy product to proliferate in a patient. And it usually consists of cyclophosphamide and fludarabine, sometimes different combinations and different drugs with similar potency are used. This might not be a problem for patients with really refractory and severe disease, but it is a challenge if we want to think about in the future treating patients with mild or moderate disease, or those who are very early in the disease course where their trajectory might not be as clear. You're very familiar from having heard about this in immuno-oncology that cost of CAR T-cell therapies can be enormous. These can range in the United States at least from $400,000 to up to $1 million per patient. And if we think about this, given the immense amount of potential use in rheumatic diseases and autoimmune diseases, a large societal burden at least. Over the past year, we have learned, at least in the oncology space, that there could be a potential risk of transformation in patients who received lentivirally transduced CAR T cells. And we currently really don't know what the risk for patients with autoimmune diseases is. And it's hard to quantify, but it is something we need to consider when we think about patient selection. And then probably the biggest barrier, and something where we really need to think about opportunities, is the sheer difficulty of getting access to these drugs. We're seeing this currently in oncology, where we have much more eligible patients than actually patients who receive CAR T-cell therapies. And we're seeing these therapies now coming to more and more patients with myeloma, to many more oncologic indications, including solid tumors. And if we think about the prevalence of rheumatic diseases and other autoimmune diseases, if we're thinking about multiple sclerosis, myasthenia, other disease types, we will see more and more of these bottlenecks getting worse, and we're lacking the infrastructure and niche expertise to really administer them at scale. As you have heard, the current model uses autologous CAR T cells. These are the ones that are currently in use in clinical medicine and immuno-oncology, are being advanced largely for autoimmune diseases. This requires the personalized production of CAR T cells, meaning taking our own patient's blood, shipping it somewhere, engineering it ex vivo, and then after several weeks, putting it back into the patient. This also requires cytotoxic therapy or lymphodepletion to allow these cells to expand in vivo. And there is a potential for secondary cancers. As you can imagine, because this product is made for an individual patient just for one-time use, this takes a lot of time and effort and cost. A lot of opportunity in the space, and something I'm very excited about, is the use of allogeneic cell therapies. Here, rather than using the patient's own blood to engineer, you're using healthy donor immune effector cells. So think about a donor donating large amounts of immune cells, T cells and NK cells, and then engineering those cells en masse and store them away so they can be taken out the freezer for any patient that requires a cell therapy product. There are a couple of engineering things that need to happen to make this an actual successful approach. One, you need to remove the T cell receptors of the donor. Otherwise, you get a graft versus host disease, or at least the risk of that, meaning the endogenous T cell receptors that would still be on the T cell that's engineered would attack the tissues of the recipient. You also need to remove surface HLA molecules to prevent immediate rejection of the cell product. Other companies are using induced pluripotent stem cells, or iPSCs, as an unlimited resource to grow up T cells and NK cells that already are engineered at that stage to infuse this into a patient. The benefits are obvious: immediate availability of the cell product, good opportunity for quality and batch control, and the possibility, at least, to offer these cell therapies at a significantly reduced cost. There is, on the other hand, rather than engineering CAR T cells outside of the patient, the future opportunity to engineer these cells in vivo, in the patient itself. So think of it as a way to deliver potential templates for the CAR T cell to the patient and getting them in the patient into the right immune cells, the NK cells or T cells. So this is complicated and has additional challenges. You need to specifically get the construct into the right cells. You need to not get it into other cells, and it needs to be done very safely. But if this can be effectively done at a way that these therapies are equally potent to the therapies that are used when we ex vivo engineer them, this is a huge opportunity to make off-the-shelf therapies that could be given as a small injection, maybe even in an office or an infusion center, without need of much engineering, and in that case, without need of lymphodepletion or conditioning therapy. There's another potential risk here. If you're only delivering mRNA, so that is a template that is only there for a short period of time and diluted out as the cell proliferates and naturally degraded over the next few days, this would basically allow you to express the payload that you need to control the disease, but without having any risk of T-cell malignancy, and probably because it's diluted out so quickly at a much reduced risk for adverse events, think cytokine release syndrome or other cytokine toxicities. The biggest challenge at the moment with this approach is the limited potency. Because you're only reaching a small amount of cells and the expression is rather low and short-lived, these cells do not have the same potency to control disease. But if we manage to figure this out in the future, this could be an incredible opportunity for the treatment of autoimmune diseases. So are people doing this? In the research realm, yes. I share with you a beautiful study that came out of UPenn that used an in vivo editing approach that is mRNA-based to target fibrosis, not in scleroderma, but fibrosis in the heart. And different to the CAR T cells that you typically hear about that target B cell surface molecules like CD19, these CAR T cells were engineered using lipid nanoparticles that provide a small lipid shell around the actual mRNA. And on the surface of these lipids are little antibodies that allow you to target T cells with some specificity. And the CAR that is then made in the target cell, which is the T cell that becomes the CAR T cell, expresses now the CAR and it targets a fibroblast protein called FAP. As you can see on the right here, they were able in animals to get a direct expression of the CAR in T cells, although the expression level is incredibly small if you compare this to CAR T cells that are engineered ex vivo. So this is probably where the limitation in potency comes from. But when they treated animals with this approach and induced a form of heart fibrosis in the animals by stressing them, they were able to significantly reduce that degree of heart fibrosis and scarring using this approach. And you can kind of see this here in cuts of the heart, where a lot of the red, the fibrosis, is much reduced in the animals that were treated with this in vivo CAR T cell approach. So there's promise for this, even though I do not know whether it would be suitable to achieve a deep B cell reset in patients with autoimmune diseases. But there are other opportunities other than CAR T cells that we should be aware of when we think about future opportunities in scleroderma and other autoimmune diseases. Bispecific T cell engaging antibodies have been used for several years now in immuno-oncology to treat B cell cancers. Here, rather than engineering a T cell, you're engineering a protein, a bispecific antibody, by taking two parts of two different antibodies. The binding domain of an antibody that binds any T cell, this is often an antibody that targets a protein called CD3, and then an antibody fragment from an antibody that targets B cells. This is the same antibody fragment, for example, that you would find on the surface of a CAR T cell that allows you to specifically kill B cells. When you make these small proteins that have only short half-lives, what they do is that they can actually cross-link any T cell in the patient with B cells. And when this cross-linking happens, the two form an immune synapse, and that immune synapse leads to activation of the bystander T cell to then get activated, secrete perforin and other immune reactants, and kill the B cell. So this is a way to have an off-the-shelf therapy that can leverage basically the patient's own immune system, the T cells, to go after the B cells without need of any engineering. In the immuno-oncology space, where these are used to treat lymphomas and myelomas, they have been incredibly successful. There's a large amount of these bispecific antibodies targeting various B-cell antigens currently approved by the FDA and in Europe. One of the oldest ones targets CD19 called blinatumomab. This is a small molecule with essentially the minuscule parts or the limited parts of an anti-CD3 antibody and an anti-CD19 antibody that are needed to get that cross-linking. And this has been very potent for the treatment of B-cell cancers. There are a number of bispecific antibodies that have slightly different formats. More and more, we see the use of Fc domains and full antibody designs that prolong half-life. And they similarly have been quite potent for the treatment of B-cell cancers. And then for myeloma, a form of plasma cell malignancy, where the target antigen CD19 and CD20 is often no longer there, the use of antibodies, bispecific antibodies targeting BCMA, has been quite remarkable. So depending on the autoimmune disease, you can see that these tools have already existed and are well used in the space in immuno-oncology and offer immense opportunities for the treatment of patients with autoimmune diseases. The question might come up, but are these bispecific antibodies efficacious enough? Cannot be as efficacious as an engineered T cell. Well, I think at least in cancer, they have been surprisingly potent. So even though it is hard to compare side by side, even in cancer, where you need to get rid of every single last B cell in order to get disease remission and a cure, some of these patients achieve that just with the use of bispecific antibodies. And the numbers are very similar to what you would expect in patients treated with CAR T-cell therapy. So what about autoimmune diseases? Well, this field is moving so fast. And just in the last couple of months, we saw 2 papers come out that started to use existing bispecific antibodies, in both cases blinatumomab, the anti-CD9 targeted small bispecific antibody that I mentioned to you, for the treatment of autoimmune diseases. In the first study, it was actually a patient with scleroderma who was treated with it. And you can kind of see the timeline on top here, where the patient received, after many other drugs, 3 rounds of blinatumomab. 4 rounds of blinatumomab at varying doses, dose escalation up. What you can see that after these rounds or cycles, the number of B cells in the peripheral blood, this is here the yellow line, went from around 1,000 to undetectable. Just after a week, the B cells came back up. So in the second cycle, after the patient was treated again, the B cells went down to 0, but it again came back up. What is interesting here is the question of whether we can achieve a deep B cell reset using this, and to learn what doses would need to be used to achieve that. Clearly, you can use large enough doses in immuno-oncology to fully control the cancer. So that's the opportunity here to learn how to do this in patients with autoimmune diseases. In this example, the patient got remarkably better, even though there is very, very short follow-up. But what you can see that after every cycle here, at least the peripheral B cells are coming back up. So this is maybe not the same depth of B cell depletion that we have become used to from patients receiving CAR T-cell therapy. Just a couple of days later, a second paper, a beautiful paper, came up about the use of bispecific T-cell engagers, in this case, again, blinatumomab, in patients with rheumatoid arthritis. In this study, patients received 2 doses of blinatumomab at different days and were administered to get a dose of 9 micrograms per day. In these patients, you can see that there was, as expected, a significant reduction of CD19 positive B cells in the peripheral blood, not a complete elimination, but a dramatic reduction. The patients also had significant improvement of their symptoms over a course of just 12 weeks. Improvement in tendon joint count and significant improvement in their swollen joint counts. So their joint disease got much better. Similarly, if you image these patients, you see a significant reduction of the amount of inflammation that's seen here. Even though the question remains whether an immune cell reset can be seen in a deep B cell depletion, these patients had a reduction, but not a complete elimination of autoantibody levels. So it remains to be seen whether these patients were able to achieve a deep B cell depletion that would lead to an immune reset. And I think we just need to learn how to use this drug more effectively in patients with autoimmune diseases. But the first use of these novel bispecific antibodies in patients with autoimmune diseases are incredibly promising. So why am I excited about this? What are the benefits of bispecific antibodies over CAR T cells? I think there could be several that make them highly suitable for the treatment of autoimmune diseases. As antibodies, they're off the shelf, either infusible or injectable. Because we're really good at giving essentially proteins and antibodies to our patients, even though this is a slightly different way and these patients still need to be monitored for side effects like CRS, cytokine release syndrome, or other cytokine toxicities, these are much more scalable. They're available at significantly lower cost than engineering a T cell, and they would allow for sequential treatment or retreatment if the depth of depletion is not achieved. And I think there's a lot to learn in this space. What is potentially most exciting about these therapies, thinking about the future of this space, is that there is no need for conditioning therapy. So the patient would not require the use of cyclophosphamide and fludarabine. And because you're not engineering a cell and integrating anything in a T cell genome, I think the risk of T cell cancer should be not increased. So if we consider this, at least theoretically, this would make these therapies much more applicable to a much larger number of patients, particularly in an autoimmune disease population. And maybe that's what is needed to get these therapies to patients with early disease, early scleroderma, and not just severe refractory disease, but also mild to moderate disease. Beyond the use of B-cell depletion strategies that eliminate all B cells, and we just touched on CAR T cells and bispecific antibodies, there are also antibody drug conjugates, there are CAR NK cells. There is a lot more in the space that we don't really have time to talk about today. But the elimination strategies that we talked about are mirrored by equal efforts in a tolerization realm. So the development of therapies that could potentially achieve an immune homeostasis against by using tolerogenic approaches rather than elimination approaches. And on the elimination side, something that I'm very excited about and my lab is pursuing is the development of precision or antigen-specific therapies that would selectively get rid of drivers of the disease rather than all B cells. So in that realm, there's a lot of movement. Rather than depleting all B cells, different approaches are being advanced to selectively target autoreactive B cells. So rather than targeting 100% of B cells, you would target the 1 to 2% of B cells that actually drive the disease. CAR T cells have been advanced for several autoimmune diseases. My lab is developing a strategy called catcher T cells that use re-engineered T cell receptors for different diseases, including antiphospholipid syndrome. And for other diseases, including scleroderma, the use of T cell targeted therapies might become very attractive. To summarize, I think there's a new world of opportunities for scleroderma. The evolution of CAR T cell therapies in itself is incredibly exciting, and I think they'll become safer, more off the shelf, and more scalable. But there are also many antibody-based immune cell engaging approaches that really promise similar potency to CAR T cells, but without the need for cytotoxic therapy, and maybe without the risk of any T cell transformation. Antigen-specific therapies, I think, are the future, and they offer opportunities to really selectively eliminate autoreactive B cells and T cells, the ones that cause the disease, without increasing the risk of infection. So I think there's a lot of hope in this space. I want you to take that away, but I think we still have to overcome many challenges to bring these therapies to our patients. Thank you again for the invitation. It was a pleasure to speak to you again, and looking forward to the panel discussion.
Well, thank you very much for the kind introduction and the opportunity to speaking to you today with the Scleroderma Foundation. Luke, it's been great, the partnership with you. My name is Peter Maag. I'm the CEO here at Kyverna. And you can tell by my accent that I'm actually German. So I'm German talking about CARs. Now, obviously, we're talking about CAR T cell therapy. And that you can see that on our very first page, patients are absolutely at front and center what we do at Kyverna. I actually wake up every morning and the first email that I read is, how are our patients doing? It has taken Kyverna 5 years to become that overnight success. We thought about bringing CAR T cell therapy to the patients with autoimmune disease, to patients like you. And we have made tremendous progress now, having focused on 50 patients and have a wealth of experience that I'm happy to share with you in the following slides. So Kyverna has been at this CAR T cell therapy for the last 5 years already. And the question that we are following is an immune set possible can we reset the immune system? And here you can see this with a very simple paperclip at the computer. You might be able to reset this computer with a simple paperclip. And the Kyverna construct, in a way, reminds me of that paperclip with the immune reset. Now, this is a paradigm shift in medicine. Many of you are taking many medications, small molecules with tablets, or then infusions with monoclonal antibodies. But really, CAR T-cell therapy promises to be that new paradigm shift in medicine for the next 30 years. And Kyverna 101, our lead product, is one of these CAR T-cell therapies that we are proposing. We have a very rich pipeline at Kyverna focusing on multiple autoimmune diseases. And I highlighted here for you the systemic sclerosis clinical trial that we have in phase 1, phase 2. And this trial is actually recruiting, and we're actively looking for patients that are engaging into these clinical trials with us. Now, the 50 patients experience, you see we have that in neurology, in rheumatology, and then in many other diseases. And the promise that we are supporting is that CAR T-cell expansion, are these cells actually expanding? Are they depleting the bad B cells and the B cells? Are we reducing autoantibodies? And do we have disease impact? And obviously, the long-term data is still yet out, but is there durable treatment response? And the first patients that have been treated with CAR T-cell therapy are now 3 years out, having this durable treatment response that we have as our goal. I'm very happy to report that actually, as I give this presentation, we have Denise, our first patient that has been dosed with Kyverna 101, 1 year anniversary. She's doing really well. It's actually just yesterday that she had her very first year anniversary. So very exciting times for Kyverna, and she's doing very well. She's a myasthenia gravis patient. I would love to acknowledge, obviously, as we're creating this Kyverna village, the patients and their families for their courage and their trust. It obviously takes a village to be successful in CAR T-cell therapy, and we at Kyverna are building that village. We would also like to thank the caregivers and their collaborators for their partnership, and the Kyverna employees that are very dedicated of bringing CAR T-cell therapy to patients with autoimmune disease. If you have any questions or more need for information as we are embarking into this journey together with a paradigm shift in autoimmune disease, please contact me, pmaag@kyvernatx.com, or contact us at Kyverna. You'll find us on kyvernatx.com. Thank you so much for your attention.
Hello, my name is Dave Shook, and I'm the Chief Medical Officer at Nkarta. I'm also a practicing pediatric bone marrow transplant and cell therapy physician, and I appreciate the opportunity to speak. These are my disclosures. So cell therapy may challenge the way that autoimmune diseases are treated. Autoimmune disease is certainly a major unmet medical need, with an estimated 7 million patients in the US with some form of B-cell mediated autoimmune disease. And B cells mediate those systemic diseases by a combination of internal and external factors. And the effectiveness of current therapies is inadequate and often consists of lifelong immune suppression. Recently, CD19 targeted cell therapy has shown impressive results even in patients with severe autoimmune disease. And in some ways, the early results with cell therapy in autoimmune disease may exceed those responses seen in cancer. Nearly every patient with scleroderma, lupus, and myositis have responded to CAR T-cell therapy, while only about half of patients with relapsed aggressive lymphoma have had a complete response. And multiple factors may account for the reason why this is. One, B-cell cancers tend to have escape mechanisms to prevent them from being targeted. They also surround themselves with a tumor microenvironment, or cells that keep them from the immune system finding them. And there's many more cells to be killed. CAR T-cell therapy for autoimmune disease is also far different than it is for cancer. We know in B-cell cancers, CAR T-cells persist long after expansion. Patients are pre-treated with 2 different chemotherapies, called cyclophosphamide and fludarabine, often abbreviated Flu-Cy. And after that, those CAR T cells proliferate or expand. Then they exist in a patient's blood for about 18 months after they're infused. And we know that those CAR T cells last longer in patients that respond to these therapies. In contrast, patients with autoimmune disease, the peak occurs similarly around day 10, but after the same amount of lymphodepleting chemotherapy, but the B cell suppression is far shorter. That means that the cells don't have to exist in the body as long. I'll highlight now a product that we're developing, NKX019, which is a CD19 CAR NK cell or natural killer cell. Unlike CAR T cell therapy, this is allogeneic, meaning it's made from another person's blood. It's available off the shelf and on demand. There is no patient apheresis and no delays due to manufacturing. In addition, there are limited additional medications, including fludarabine, antibodies, or cytokines. And this drug has been successfully given to dozens of patients with cancer, and we know that this can be given completely as an outpatient with an excellent safety profile. The cartoon here shows NKX019 as a CAR NK cell. It has two main components: the CAR, which targets CD19, and the membrane-bound form of a cytokine called IL-15. This allows the cells to live in the body longer. NKX019 is given 3 doses after a single dose of cyclophosphamide. And lymphodepletion with cyclophosphamide, as opposed to cyclophosphamide and fludarabine, may lessen the risks associated with cell therapy. CAR T cells have a peak expansion, as we discussed, at around 10 days. That 10-day, that occurs after fludarabine and cyclophosphamide combined. And this addition of fludarabine increases the risk of prolonged cytopenias or low blood counts, infections, and secondary MDS. Patients treated with CAR T cell therapy have approximately 4% risk of cancer that's associated with the lymphodepleting chemotherapy, as well as an FDA risk of T cell malignancy. NK cells, in contrast, are immediately active and do not require expansion and get the same amount of levels in the blood with cyclophosphamide alone. This short-lived NK cells may limit the risk of prolonged B-cell aplasia and secondary cancer. In summary, NK cells may be the ideal cell therapy for autoimmune disease. They reach peak activity at infusion for rapid activity without in vivo expansion, with potential for similar activity as CAR T with a much improved safety profile. They have a low risk of expansion-related toxicities like cytokine release syndrome or neurotoxicity, and the elimination of fludarabine may reduce toxicities for patients, including cytopenias, infection, and DNA damage that can lead to cancer. And the superior safety and accessibility of NK cells prioritizes the patient experience, the on-demand availability without need for cumbersome infrastructure at sites, need for patient blood collection, and treatment delays. And this allows this therapy to be given completely as an outpatient. Reducing toxicity associated with cell therapy will allow more patients to be treated, especially in diseases where safety is critical. Thank you.
Well, great. I wanted to welcome everybody to the Q&A session of our CAR T and advanced biologics session today. I think I hope I speak for sort of everybody, and certainly everybody on the panel. I think this is the absolute dawn of an era potentially offering really curative therapy for a range of autoimmune diseases, including scleroderma. Obviously, it was a whirlwind tour over the last half hour, so I want to just give maybe a couple more minutes to go back in and fill in some of the points that I think the audience would appreciate. Let me ask Dr. Merkel if he would just provide a little bit of perspective on some of the longer term results, such as we have them, both from academia, which is largely the longest term experience. Peter, please go ahead.
Yeah, hi, thanks, Luke. This is a Terrific program. I think we're all very excited about the potential for cell therapy and especially CAR T19 for the treatment of scleroderma and other autoimmune diseases. The data we have really get very much less so as we go further out. So the initial information we have are for patients with lupus, particularly the longest term out is a couple of years from the first young woman who got this therapy for lupus. And now it's beginning to accumulate. What I can tell you from seeing the data from Georg Schett and some data from China is that what we're seeing is a fair amount of consistency. That is, we're seeing for the most part, the therapies are effective, but not everybody. Remember, no therapy that we know of is good for everybody in every situation. So I think we need to be careful. And we're still learning. These are experiments. But we're seeing that these long-term outcomes are quite good therapeutically and quite good from a safety standpoint. What I think we're particularly interested in from a safety standpoint is that there's a rapid return of the B cells, which are the cells that are eliminated with CAR T, and that people's immunoglobulin levels, which are these proteins that help us fight infection and are also what we stimulate when we get vaccines, not only do they not necessarily go away, but they come back to good levels. And I think that's really important. And from an efficacy or does it work standpoint, we're seeing each time we see new updates, longer term benefits, where most of the patients either got better, didn't get worse, but importantly, aren't back on therapies that suppress the immune system. That is not always the case. That depends on the situation. And I'll remind you that we haven't tested it in that many patients, and it hasn't been done in people who are very sick or maybe very well. So it's I'm measuring my excitement for this whole field with the fact that we're still doing experiments properly done in trials that we and others are running.
Thanks, Peter. Just maybe to contrast that, or just maybe just to offer a little bit of comparison, you did a phenomenal job with the review of stem cell transplant with chemoablation. What's been the success rate in scleroderma overall? You referenced it, but just to put that in people's mind again.
I think it's hard to get a number on that because sites report intermittently, and there's different attitudes towards this. In general, I think the feeling is that many patients are doing pretty well. There have been some significant patients who do not do well. Either they have significant toxicity or their disease continues to progress. So I think we haven't seen perhaps anecdotally quite the response we've seen in some of the trials. But I remind you that for myeloablation and autologous stem cell transplant, when we do that, they're highly selected patients. So they're really very selected patients. So I think we've had some people who've done well and some who haven't. That's a very wishy-washy answer because it's hard to get our head around it. The numbers are not huge, and it's a little different in Europe than in the US for who they're treating. So I would hesitate. It's in the neighborhood of doing well for some, but remember, we're selecting people very carefully.
Thanks. Peter Maag, you talked obviously about Kyverna 101, autologous CAR Ts. Maybe you just remind us what that therapy is. I think Max went through it and I think did a masterful job, but just to sort of put it in everybody's mind, what sort of the steps are, and in particular, I think it's useful to talk about some of the conditioning regimens so that I think we're going to line up some of the complexities of each of these therapies when we get to the end of this conversation. I think you're on mute.
Look, first of all, thank you so much for having me on this call. It's so important what your foundation is doing, so thank you.
Thank you.
In terms of CAR T cell therapy, our Kyverna 101 is really using the power of your own immune system. You're using your T cells. We isolate them through an apheresis procedure. We then, what I call, put a hook onto it, and I'll demonstrate this always with a simple paperclip, because like the paperclip that I'm holding here, the Kyverna construct looks like this paperclip that we add into your CAR T cells and then reinfuse that changed CAR T cell into you as a patient, and that hook finds the pathogenic B cells or B cells and depletes these B cells by hooking onto the B cells. And so far, we have now treated 30 patients,
that is working extremely well in patients with various autoimmune diseases, because these B cells are producing autoantibodies, as you might know, and then these autoantibodies are causing all kinds of issues in patients. The key question is, how long do we need to deplete these B cells, and how long until these autoantibodies might be coming back? So to Dr. Merkel's point, the Kyverna 101 construct has been constructed with safety in mind. It's still an experimental therapy, so we need to be very thoughtful. But having treated the first 30 patients with Kyverna 101 allows us to now gain experience. And just 2 days ago, our very first patient at Kyverna was 1 year out. She's a myasthenia gravis patient, and she's completely free of disease, which is extremely exciting. But I would say, you know, the results will be varied, because we do treat patients that have tried many different treatments and now ended up in a CAR T-cell treatment at this stage. And so we need to think about how this paradigm shift will be impacting many patients in the future.
Yeah, thanks. Max, I thought you did a really nice job of discussing sort of a range of therapeutic opportunities and talking a bit about some of the conditioning regimens and the differences between those two, or among the different, multiple different opportunities. Maybe just to focus on T-cell engagers for one more second and highlight, you know, what some of the differences are for patients. Do you think there are going to be different patients who are going to qualify in for a T-cell engager just because of the conditioning regimens, or how do you think of the future of cell therapy versus biologic therapy?
Yeah, such a good question, and thanks for having me. This is really great crowd to be part of. We're in a really opportune situation that, you know, we in rheumatology can kind of like shift and peek over to immuno-oncology and leverage a lot of the therapies over the last 10 years or 20 years that have been developed to target B cells, malignant B cells. And because you have to cure cancer, really get rid of every single last B cell, these therapies are very potent, more potent than anything we have ever had in rheumatology. So CAR T cells are the first of these therapies that have kind of spilled over into our world. And we're seeing, you know, the transformative effects that they can have on diseases like lupus and maybe also scleroderma. But there are other ones out there that can equally be repurposed. And I think the theme of the most potent therapies that we're currently have at our disposal is that we're leveraging the patient's own immune system, or the immune system, to target the bad actors, the B cells that cause the damage in a given disease like scleroderma. You can do this by, as Dr. Maag said, put a hook on a T cell and reinfusing that. But there are other options where you can just infuse a protein. Think of like rituximab that we've given for many decades now, where you go after the B cell and put a hook as a protein on the B cell that then grabs another T cell that's already in the patient and kind of brings it close to the T cell to activate that T cell to kill the B cell. So you can do this by engineering either a patient's own T cells, or you can do it by using these engaging molecules. There are some benefits to the engaging molecules, but I think time will tell what the right approach is for what patient. We know that CAR T cells can do a really good job depleting very deeply the B cells. And that's what people think of when we talk about immune reset, that very deep B cell memory compartment depletion. For the engagers, we know from immuno-oncology that they can also be curative in cancer. So they have a very similar potential, at least in terms of potency. But we still need to learn how to use them best in patients with autoimmune diseases. So I think from a T-cell engager perspective, we will eventually learn how to use them just as effectively as CAR T-cells. I'm not sure that we're there yet. But what is, I think, at least encouraging is if you think about, you know, the spectrum of disease, patients who are very refractory and patients who might be much earlier in their disease and might be, you know, less willing to take on the barriers of conditioning therapy, might be worried about risk of transformation, other risks that you might have in mind. Something that's off the shelf and can be given without conditioning therapy is very attractive. I think at the moment, the FDA thinks of it as kind of the same class of drugs, but I think we'll see potentially these become a spectrum between milder disease and more severe disease, and we'll learn to apply these drugs to what patient population.
Thank you. That's really helpful. So let me then just sort of weigh in here with Dr. Shook. So Dave, we talked a little bit, obviously you're featuring allogeneic therapy, an off-the-shelf product. Do you want to talk about how the protocol ended up using exclusively cyclophosphamide? Obviously, you did reference the fact that you were able to drop fludarabine. So is there some clinical evidence that that level of chemoablation leads to an equally deep immunological reset? I think maybe just helping everybody understand how that conditioning regimen evolved would be helpful. Thank you.
Yeah, sure. And thanks for the opportunity here. I think the reason why the lymphodepletion regimen is different is because the cell type is different. I think we talked about the T cell with the hook or the paperclip, and we talked about the bispecific T cell engagers, et cetera. I think where we feel like a middle ground is to have the power of a cell therapy and the convenience of an off-the-shelf therapy together, and to be able to link that. And T cells, as I explained before, are infused at kind of a low dose. We have a big expansion after a few days, similar to how we feel a bit worse a few days after we get a flu shot or if we get exposed to something. And that expansion process takes a bit, and it takes a different kind of environment to happen. With a therapy like NK cells, those cells don't really expand or duplicate themselves, replicate themselves once they're infused. So they don't need the same kind of lymphodepletion. And fludarabine is really important for that cytokine induction, or that making the body release those chemicals and to suppress the immune system for a long time. And so we feel that the priority, certainly among patients, and people living with these autoimmune diseases, is safety, right? I think that is the priority here, and so we believe that reducing the amount of exposure of chemotherapy is going to be critical here. And I would agree with the rest that generally the therapies themselves, the targeted therapies, the T cells, the others have been relatively safe over a long period of time. The chemotherapies are not safe over a long period of time, particularly fludarabine and others. I'm a practicing pediatric oncologist right now, and we fear every time we give these medicines because, like patients, people living with autoimmune diseases, those children have hopefully 10, 20, 30, 40 years left to live. And so when we have to contemplate long-term side effects, so it was intentional to try and eliminate, or at least to reduce the risk of, wanted the barrier to access these therapies not be safety.
Thank you. Let me go back to Dr. Merkel and talk a little bit about patient eligibility and some of the current clinical trials, which I guess portends a little bit, the direction of availability for medicines should they, reach the marketplace. Dr. Merkel, please.
I guess so. Eligibility in terms of who would be eligible for a CAR T for scleroderma, for example. We're actually opening a trial in about 2 weeks. We're opening our trial that will include scleroderma here at the University of Pennsylvania. We'll take people from all over the country. And what we're doing is, I hate to put it this way, but we're looking for sick but not too sick. We're looking for people who have enough scleroderma that's been active and ongoing that requires therapy that we are worried about is going to progress. So we don't want people who have had 10 years and it's not as active, even though it may have really impacted their lives greatly. And we don't need people who are too sick because we want to make sure that it's safe at first. So as we get more and more, if this is effective therapy, we're going to go to sicker patients, and we're probably going to go to less sick patients as we expand it. But right now we're looking in the middle, we're looking for people who have scleroderma that's been active with skin disease, that they could have some lung disease, they could have some other things, but not too far gone into the course of their therapy, and that they're safe enough to be able to get the therapy. So some skin disease, maybe some lung disease, we have some criteria that will work it out, because we want to make sure that it's safe to go forward. And they should have been on at least one medication in the past, and it's still active even though they've been on a medication. Unfortunately, that's a lot of our patients. So we think that there will be people who will be interested in taking this. And I remind people, this is a option, but it is an experimental option. So I think we're happy to screen people who think this may be something for them if they want to participate in research.
Great. Yeah, we're down to our
What I
last
would add
minute.
is that the specifics for any individual are very important. And so, we're very careful.
Yeah, we're down to our last minute. Let me just maybe ask Dr. Konig to talk a little bit about sort of the cost. And we only have 60 more seconds, but just to talk a little bit about sort of the availability from an economic perspective and how that might impact some choices that get made as we go forward.
Yeah, I'm obviously not an economist who can model this, but these therapies are expensive. I think we all realize that. I like to think of it as, it used to be incredibly expensive to sequence a human genome, and now we're 20 years later and everything is dirt cheap. So yes, at the moment, these are expensive. They are much more expensive than conventional drugs, but they can have profound effects. So I think somebody who's more qualified will be able to make an economic argument that this over the long term, over 10 or 20 years, is actually cost effective. That said, I think there are new modalities that the field is going to gravitate towards to make it more economical. And I think these are off-the-shelf therapies, whether that's a protein engager or whether that is an allogeneic cell that can be produced at larger scales without having to make a product for each individual patient. But I want people just to understand that what we're using now is not what we're going to use in 5 or 10 years, right? This is going to be a moving field. We'll learn as a group, and it's only going to get cheaper and likely more effective.
Well, I just want to thank everybody for listening in and spending an hour of your time with us. Also want to thank our illustrious presenters, Dr. Peter Merkel from Penn, Dr. Max Konig from the Johns Hopkins Institute, Peter Maag from Kyverna, and Dr. David Shook from Nkarta. Thank you all for participating. It's been incredibly illuminating, and we're looking forward to the future of this therapy for many of our patients.
Thanks.
Bye for now.
Thank you.